Hi, we're looking at chapter 14. We're going to begin by reviewing a little bit about the DNA structure, what kind of things it's made out of, and some of its features. And then we're going to spend a little bit more time talking about the more complicated process of DNA replication. Basically, how does DNA go from being one strand to two strands? During the S phase of the cell cycle, what is going on? How is all that genome being copied? And then the last part of this lecture, we'll talk about some of the things that can go wrong when DNA is not quite copied right, as well as some of the ways that DNA can be repaired if it is copied wrong. For starters, though, I like to back up and get a little bit of perspective. What you're seeing here is just a picture of a skin slide. So this would represent like a little pinprick of your skin. This is probably about how many cells would be, if you maybe just took your little pencil and touched it to your skin, that amount of region that you're touching would probably be about this many cells if you were looking on a microscope. And every single one of these cells has got a little nucleus inside of it. Okay, so each one of these things here are the cells. Inside those tiny, teeny cells are these little nuclei, and inside these dark stained purple nuclei is all of the information required to build your entire body. All of the information to build your entire body is right here. It's also right here. It's also right here. It's also right here. So this is just a mind-boggling amount of information inside a very, very small space. So for starters, that should just blow your mind. But there's going to be a couple other interesting facts here about DNA, which I think is kind of fun to talk about. This is all just FYI stuff. You don't have to take notes on this. This isn't going to be on an exam. It's just to kind of give you some perspective here about how amazing DNA really is. So it codes for all the information required to build you, and despite that complexity, it's really only built with 4 letters. Okay, so if you imagine DNA being like a text and the instructions of how to build something, we have a 26-letter alphabet that can make a whole bunch of words. Well, this alphabet is only 4 letters long. Basically, it's different combinations of these 4 bases. So the letters are A for adenine, G for guanine, T for thymine, and C for cytosine. And depending on the series and the arrangement of these letters, it dictates what is going to be built. 99% of our DNA is exactly the same, so my DNA is literally almost identical with all of your DNA, save just less than 1% is different between you and I. So we're pretty much exactly the same. A couple of differences here and there that give us some unique features, but I mean, when you think about how cells are running and like all of the organelles in a cell and also all of the different organ systems and things like that, I mean, it's very similar between all of us. If you could type 60 words per minute, 8 hours a day, it would take 50 years to type out the genome. Okay, these are some of my favorites here. All right, so if you were to take out the genome of one cell, so if I took just one single one of these little skin cells, took the DNA that's located in the nucleus here and unwound it, stretched it out so that it was all of the chromosomes were linked up into one single file line of DNA, it would be about 6 feet long. Okay, so 6 feet of DNA fits into one cell. Well, this is the crazy thing. There's like 100 trillion cells in your body. So if you were to take all of the DNA from all of your cells and line it up end to end, it would go from here to the sun and back again 600 times. That is a super long amount of distance to travel. That is like outrageous to me. I don't know, maybe this doesn't boggle your mind as much as it does me, but think about developing in your mother's womb. And think about this sheer distance from the Earth to the Sun and back again 600 times. And think about that amount of linear feet of DNA being made in the span of 9 months as you're developing in your mother's womb. It's just like there is just an insane amount of DNA being built, and there's just an insane amount of DNA inside you. And all that information in one genome can be packaged up and fit into one cell. I'm never going to be able to understand that. That's crazy. There's a pretty good link here that gives you some illustrations just about how much information this is. They actually took the effort to sequence a person's genome, meaning they could go in and see what all of the series of the letters of A, G, T's, and C's were, sequence the whole molecule of DNA, and then print it out page by page, book by book. So it's kind of fun to see the visualization of just how many books it would take to hold the information of somebody's genome. So feel free to click on that link if you like. Again, that's just for fun. It's a pretty entertaining video. I recommend if you have some time, you watch it. All right, moving on ahead then to the structure of DNA. This is a little bit of review. It's a double helix, which means it's this shape. Okay, there's two rungs on either side, and they spiral together. So if you again imagine kind of like a ladder that has different rungs in it, and then you were to twist that ladder so it was a spiral shape, that's what a double helix would be, and that is the shape of DNA. DNA has two sides to it, right? So this, just like a ladder's got two sides to it, DNA has two sides to it. So this would be kind of one strand, and this would be another strand, and then basically there's a whole bunch of these bases, these A's, these T's, and these C's, and these G's on one side, and they're actually bound to a bunch of bases on the other strand. And so right here we would call these base pairs, okay? Because you have one, what's called a nitrogenous base over here, and that's attached to a nitrogenous base here. Now the thing that, so the base pairs are in the middle of the molecule, and then on the things that make the support, kind of like the sides of the ladder, if you so to speak, this is called a sugar phosphate backbone. And this is what gives the DNA molecule its structure. You have alternating, there are alternating sugars, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, all linked up to create kind of this structural material. And then the series of bases in the middle are going to be what kind of codes the information. Speaking of base pairs, the different bases we have are A, T, C, and G. Well, when we're talking about how these pair together, A always pairs with T, and C always pairs with G, and vice versa. So if there was an A on this side, there'd be a T over here. If there was a T on this side, there'd be an A over here. If there was a C over here, there'd be a G over here, and hopefully you kind of get the picture. So DNA is made out of nucleotides, and nucleotides have three parts to them. The first, and we've already just talked about this, but the first is they have a nitrogenous base. So we said there's A, G, C, and T. Here's some pictures of what they look like. Then there's also going to be the phosphate group and the sugar. So here's kind of imagine that this is one strand of the ladder, and this is the other strand of the ladder, and we're looking at it molecularly. So let's say we kind of took a small piece and we're looking at the actual molecular structures. Here's a phosphate, here's a sugar. Here's a phosphate, here's a sugar. So all of this together forms the sugar phosphate backbone. And that's also going to be found on the other side. There's a phosphate and there's a sugar. And there's a phosphate and there's a sugar. So this is called also the sugar phosphate backbone. So these are the two sides of the ladder. And then in the middle are the base pairs. So the nitrogenous bases that we're looking at, there's thymine. Thymine always pairs with adenine. So here's a base pair, two bases on either side of the molecule. And then here's another example. We have guanine, and that's paired with cytosine. The thing that's holding these two, the thing that's holding this side of the ladder with this side of the ladder is these little dotted lines which represent hydrogen bonds. So that's what's keeping these. And these they're weak enough that they can be broken apart or realigned back together. And that's going to be kind of one of the secrets to how DNA is replicated. So a nucleotide, when we say it's a nucleotide, it's got these three components to it. So if, like in this example, this whole series of molecules here would be one nucleotide because we have the phosphate, the sugar, and the base. And then here would be another one, the phosphate, the sugar, and the base. And then there'd just be a repeating, very, very long repeating segment of these, so long that it could, if you added all of them up in your body, it would go from here to the sun and back again 600 times. All right, so lots of DNA. one of the other things that's interesting here is the sugar. Keep in mind that when we're talking about this, we're saying it's a pentose sugar. Okay, so that's one of the three components. And we say it's pentose because it's got 5 carbons. This word pent is the kind of the prefix for 5. So here they are. There's 1, 2, 3, 4, and then 5 would be here. And keep in mind, in a diagram like this, the carbons aren't actually shown, the carbon atoms. They're just assumed anywhere where there's a point. It's just a way to draw these molecules. Okay. All right, so and then specifically the pentose sugar when we're talking about DNA is deoxyribose. Okay, that is the name of this specific sugar that's found along the sugar backbone. If this was not DNA but instead was RNA, then it would not be deoxyribose, it would be ribose. And there would actually be one extra oxygen molecule hanging off of it. Okay. Another feature about the molecule of DNA is it's called antiparallel, and what this means is that the two strands are running in different directions. Okay, so here's our ladder, and this is what it looks like when it's unspiraled, and then this is what it looks like when it's spiraled. Well, there's these designations we give the sides of the DNA, and that's what these things are here. It's 3' and 3' is what it's called, and 5'. That's what these little dashes mean. They mean prime. I don't know exactly why they name it that way, but that's just what we call it. So this is the 3' end, and this is the 5' end. And notice that the 3' end and the 5' end are running this way in this ladder, but over here the 3' end is actually down here, and the 5' end is up there. Okay, so they're facing different directions, and that's what we mean by antiparallel. Means that the different strands on DNA face different directions. Or maybe you could say, maybe face isn't the best word, maybe you could say that they run in different directions, and specifically these are the strands of DNA. Each strand on either side, okay, are running in different directions. Now, where we get these numbers, 5' and 3' or 3' and 5', that gets back to this picture here, okay? So the way that we name these carbons, this is called carbon number 1. Usually the first one that's next to the oxygen is carbon number 1. So this is carbon number 1. This is carbon number 2. This is carbon number 3. This is carbon number 4. And then this is this one that's right here at this bend angle. This is carbon number 5 in the molecule. Okay, and you can see that the third carbon, the one here, is the one that's always facing towards this direction, right? So that's why we call this the 3' end, whereas the 5 carbon, number 5 carbon, the 5th carbon is always facing towards this direction, so we call it 5'. So it really just depends on when we're looking at the sugar which carbon is facing which. And I'll give you a little hint. If you imagine that these pentose sugars are this little, where the oxygen is here, this forms like maybe kind of like a little arrow or a little point. The point always points in the 5' direction. Okay, so here's this 5' is this way, and then this one 5' is this way, and you can see that these are almost like little arrows that point in the 5' direction. Now, this is actually going to be incredibly important because when we talk about DNA replication, the fact that this is antiparallel throws a bit of a wrench into the whole process. And so in order to understand why the DNA replication has to occur the way that it does, we do have to keep in mind this antiparallel nature. So make sure that you understand what this means and kind of a little bit about these 3' and 5' things here before we progress and talk a little bit more complex manner of DNA replication. All right, so the basis of DNA replication, before we get into the actual process of it, there used to be basically 3 ideas of how DNA perhaps did replicate. The first was something like this. Here's this is what you're seeing here. This represents 2 strands of DNA. Okay, so when you see these little gray boxes here, they're basically trying to represent this. There's one. This would be represented as one gray box, and this would be represented as another gray box. The 2 strands of DNA. Okay, so if we go back here. Here's one strand, and this is the other strand of DNA over here. Okay, and there was a couple different ways that it was thought to replicate. One way thought that the original two strands of DNA were copied, and then it led to this situation where there was these two original strands of DNA were conserved, but then there was just something that copied two new strands off of these, forming the replicated copies. So when this double-stranded DNA replicated, it yielded two new double strands. One was just all of the original stuff, and the other double strand was all new stuff. So this was called the conservative method because all of this DNA was conserved here in its original state, and there was just the new copy added to it. Okay. Another method thought that perhaps that little chunks were replicated at a time, and then they were kind of embedded in there. So there was kind of this dispersal of new versus original DNA. That's what this blue stuff here is trying to represent, newly copied DNA. Okay, so that was the dispersive method, and then another method was that perhaps this strand went this way and this strand went this way, and then they each were used as a template strand to create a copy, a new copy of the other strand. Okay, so then you would still end up with two new double-stranded DNAs, but each of them have half of the original DNA and then half new DNA. And then so on and so forth. And there's a whole bunch of experiments done using E. coli and other things, and this was the model that was shown to be most likely to fit the data. So this is what we're going off here, the semi-conservative model. Two double-stranded DNA breaks in half, and then each strand from that original one is used as a template to create a fully new strand. And then these kind of become two new double strands. Right. Now, how this process actually occurs, we'll start talking about what's going on in prokaryotes because they're a little bit simpler to deal with, and then we'll talk a little bit about eukaryotes as well. But the reason why we start talking about DNA and replication in prokaryotes is because they have a smaller genome, and I should back up and say that all of this stuff that we figured out here about how DNA is actually replicated, it was discovered doing a lot of studies using the bacteria Escherichia coli, or E. coli. You've probably heard of this name before. These are some pictures of E. coli bacteria. They have kind of a rod shape to them, and they're very common bacteria to be found in the colon. But in either case, why we did a lot of studies on these is because they have a relatively small genome, only 4.6 million base pairs. And this can all be copied in 42 minutes. So that's a pretty fast turnaround. And so if you have a model organism that's copying its genome pretty fast, it makes it easier to study that process of DNA replication because there's kind of a lot of trials, so to speak, in a short amount of time. And if you do the math on this, it figures to be 1,000 nucleotides of DNA are added in 1 second when you consider the speed of this process in prokaryotes. So like if we go way back here to this picture, remember these are nucleotides in a strand of DNA, and so if there's adding a whole bunch more nucleotides to this strand, 1,000 are added in 1 second. Like so right now 1,000 were added, and right now another 1,000 were added, and right now another 1,000 were added. So it's a pretty extraordinarily fast process, and this is kind of tells you why, like when we're in our mother's womb, we're able to create this sheer unimaginable amount of DNA because this process does really occur fast. All right, so enough about why we just use prokaryotes, and let's actually talk about what we learned from prokaryotes. So DNA replication, one of the biggest things that we found is there's this really powerful enzyme called DNA polymerase. And as the name suggests, a polymer is something that's made up of many units. So that would be, DNA would be an example because it's made up of many units of nucleotides. And so when you read the name here, this is a DNA polymer making machine. So this polymerase means it's an enzyme. Anytime you see this word AS, this ending on a word ASE, that suggests it's an enzyme. Okay. So this name then means it's the enzyme that builds the polymer of DNA. So this is a thing that's actually going to be adding nucleotides to a template strand of DNA. And in this picture, this is kind of an animation showing what the protein looks like. Remember, proteins can have these alpha helices and some of these beta pleated sheets. Then they're made out of amino acids. Well, here you're seeing this is a strand of DNA, and the DNA polymerase is kind of riding through it and adding nucleotides to it as it goes. So this DNA polymerase is a big enzyme here in this whole process. And in prokaryotes, there are 3 different types of DNA polymerase: polymerase 1, 2, and 3. 3 is the one that's used mostly for making new DNA, and then 1 and 2, these are more for repair type situations. And if you didn't quite catch that, DNA pol is just a shortened word for polymerase. So when you see DNA pol 1 or DNA pol 2 or DNA pol 3, just know that we're shortening this word, so it says DNA pol instead of polymerase. All right, this whole process begins at what's known as the origin of replication. You might think, oh man, a genome of 4.6 million base pairs, where on earth does DNA replication even begin? Where does the cell determine where to even start this process? Well, there's generally a region on the DNA called the origin of replication that tells the cell where exactly to start the process of DNA replication. And so this is a pretty good little figure just to show you a little bit of how this is done in prokaryotes. Remember, prokaryotes don't have a whole bunch of chromosomes like we do. Prokaryotes only have one big chromosome. And this picture is not quite sure. This is showing you kind of the one circular chromosome. It's not really showing it to you in scale, but it does kind of still bring the point home. Okay, so imagine that this is one circular, really long circular piece of DNA, and right here somewhere in the middle, this would be called the origin of replication. And what happens is in that origin of replication, the 2 strands of DNA actually disassociate. They come unbound from one another, so there's a little gap here between them. And then what happens is little RNA primers are laid. And generally, an RNA primer in this process creates a binding site for the DNA polymerase. Okay. For whatever reason, DNA polymerase, it goes and it seeks through the whole genome, and if it finds a spot where there's a piece of DNA on one side and then a small piece of RNA, not DNA, but RNA on the other side, that is going to be the thing that tells it to start there. Okay, and so then what happens is that this will proceed both ways. The DNA will unravel this way, and the DNA will unravel this way. And you can see that as it's unraveling, and basically you'd imagine, little DNA polymerase enzymes are one's gonna bind here, and let's say cruise along this way, and then maybe another one would bind here and cruise along this way. And so you're starting to see that a DNA replication is proceeding in both directions. And as it just works its way all the way around the whole circle of DNA, and then you can see that it's going to end up forming 2 new circular chromosomes where there was just one before. So as this DNA polymerase is moving, it's adding nucleotides, and it's adding them in a manner that's consistent with complementary base pairing. So anywhere there's a C on the, let's say anywhere there's a C on the old strand, it's going to add a G on the new strand. And anywhere there's a T on the old strand, it's going to add an A on the new strand. And anywhere there's an A on the old strand, it's going to add a T on the new strand. So because we have this complementary base pairing, the DNA polymerase has a rule by which to add nucleotides on the new strand. It always knows what's going to be added because it's going to add a complementary base pair. And so you can see that this little reddish color here is representing the new strand that's being built by DNA polymerase, and then it's using this old strand as a template. But then in either case, when this is all finished, it's going to be a double strand of DNA, and then this is going to be a double strand of DNA. And so you're starting off with one circular ring of DNA, and then that's ending up as two rings of DNA. And then each of these new rings of DNA that's made, one of the strands in the double strand is new, and one of the strands is what we would call the template. So this is how it occurs generally in prokaryotes. Now, how these nucleotides are actually added is kind of another story. Okay, so this is going to get us into the more lengthy process of what's actually going on molecularly here. What are the enzymes doing to make all this DNA? And that's where things get quite a bit more complex. So I'll try and just walk you through. I'm going to walk you through the process step by step based upon a figure. But keep in mind, to really understand what's going on in this process, it's the best to probably see a video of it because there's just a lot of moving pieces, and it's kind of nice to see the animation and how things are happening sequentially in video animation form. But we'll kind of introduce some of the key players here and what they do, and then I'll show you a nice animation of the process. The first thing that happens is this enzyme called helicase is going to open up the DNA. So remember, DNA is a double-stranded molecule that's spiraling around, and helicase is going to be the thing that comes by here and actually opens that double strand up. It's going to break apart those hydrogen bonds so that the double strand is now two single strands. And if this picture that's in your book, it kind of represents like what would be happening right here. So if we imagine that's what's the case, here's the picture that's in the book, and the picture's not the greatest, but it does kind of show all the things that are happening. So what this picture is trying to show you is this enzyme helicase, which is indicated by this little blue triangle here. This is splicing through the DNA molecule. There's this other enzyme called topoisomerase. That's going to be the thing that unwinds the spiral, and then the helicase is the thing that cuts through the two strands of DNA, creating two single strands. Now, these two single strands of DNA are going to want to jump right back together again right after they've been cut. So they actually get coated with these things called single strand binding proteins. They're just these little proteins that bind here that keep the two strands from coming back together after helicase has cut them. So that would be the next step in the process. The single-stranded binding proteins coat the DNA. This place where all the replication is occurring is called the replication fork. And it's called the replication fork because it looks kind of like a tuning fork. I don't know if you've ever seen a tuning fork, but it looks kind of something like this. And you like hit it on a table and it rings a certain pitch. Well, that's what you're seeing here. There's one place where there's still the original double strand of DNA, and then beyond what's called the replication fork, two new strands of DNA are emerging. So right at that point, the replication fork, though, which is right here where the helicase enzyme is working, that's where these are binding. The next thing that happens is that the primers are laid, and again, the RNA primers are just short little segments of RNA. RNA is similar to DNA with just a couple of differences. One, it has ribose sugars in the phosphate sugar backbone, not deoxyribose sugars. And then the other big difference is that RNA has uracil instead of thymine. So anywhere there would be a T, there's no longer a T. It's what's called a U. But for whatever reason, it's kind of beyond me. I'm not exactly sure why. Whatever reason, DNA polymerase likes to start wherever it sees an RNA primer. So that it kind of searches through all of the genome, and anywhere it sees there's an RNA primer, it's going to attach, and then it's going to start adding some nucleotides to a template strand of DNA. So so you can see, like right here is kind of where this is all happening. As soon as helicase is unwound, broken through the DNA, this enzyme called primase is going to bind to that region, it's going to kind of create a short little RNA primer. That's this little strand that you see that's being made. It's going to be complementary to the original strand. And then that is going to be a signal that DNA polymerase then, and this would be DNA polymerase 3, can then find that spot, attach to it, and then start creating DNA from the template strand. Okay, so here's what's the tricky thing though. DNA polymerase can only bind to a 3' end of a primer. And furthermore, DNA polymerase, it builds new DNA in the 5' to 3' direction only. And this is where everything gets all confusing. Okay, because take a look. Remember we said 3' and 5' refers to which carbon atom is facing which way. So if the third carbon faces this way and the 5th carbon faces this way. Well, here is 5' end and here's the 3' end of one strand. And then this is the 5' end and the 3' end of the other strand. So DNA, it always has to bind to the 3' end of the template, but then it builds new DNA in the 5' to 3' end. So in this case, here's the 3' end, and DNA polymerase on this particular strand, it can just go ahead and bind to the primer, and then as it runs along, it's always reading off of the 3' end, and then it's building in the 5' to 3' direction. So that's pretty simple. Now, the thing that's tricky of all this though is that on this strand over here, this is actually the 5' end. So you got to catch this here. This is the 3' end here on the template strand, but over here this is the 5' end. And DNA polymerase can't just attach to this and build from the 3' to the 5' end the new DNA. It can only build in the 5' to 3' direction. So this presents two different strands. There's what's called the lagging strand and there's the leading strand. So at any replication fork where the two strand of DNA is split in half, one of these strands is just copied. DNA polymerase just cruises along and it follows right behind helicase and just copies all the new nucleotides to the template strand. But on the other strand, which is called the lagging strand, DNA has to be built in segments. So what happens is that there's a primer laid, and then DNA polymerase will actually build it in this way. And then once the helicase is continued on, another RNA primer will be laid, and then DNA polymerase will build another short segment. And then as the helicase continues, then it will build another segment. And so in this way, it is in fact building new DNA in the 5' to 3' direction, but it has to be done in short fragments of DNA, which are called Okazaki fragments. All right. So the lagging and the leading strand will be created, and then there's a problem because we have all these discontinuous pieces, and we have all these little RNA primers scattered around here. So I don't know if you can quite see this, but anywhere there's a little orange piece here, that would be an RNA primer. And then the red here represents DNA. So we have to remove the RNA primer, and this is removed by this thing that's called an exonuclease. This is actually just a region on DNA polymerase that removes RNA. Okay, so this DNA polymerase is a kind of a tricky enzyme. It not only can add new DNA to a template strand, its other function is that it can actually remove RNA. And when it's doing the removing, it's called exonuclease activity, because exo means to kind of get rid of, and nuclease refers to nucleotides. So this enzyme literally gets rid of nucleotides. This is more of something that's done on the other DNA polymerase molecules. So DNA polymerase 3 doesn't so much have this capability, but some of the other DNA polymerase enzymes do. But that's what needs to happen. These little RNA primers have to be kind of taken out, and they're taken out through this exonuclease activity of the other DNA polymerase molecules. Okay, then those little small regions that have just been removed, now there's a big gaping hole in this DNA molecule, and so DNA polymerase is going to have to attach and patch those back up with DNA now, not RNA. And then the last thing that happens is that there's going to be all these little discontinuous chunks where all of the pieces have been added and taken away and removed and added. So all these little chunks have to be stitched up together to form one nice continuous long strand of DNA, and that's done by the enzyme known as DNA ligase. Okay. So here's kind of what it looks like in animation form, and this is probably the most helpful in order to understand what's actually going on.
DNA is a molecule made up of two strands, twisted around each other in a double helix.
Twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Well, there you have it. One concise 3-minute video to explain kind of this entire part that we've talked about thus far. So here's what you want to be able to do, especially for the exam. There's a series of important enzymes involved in this process, and you need to understand what each of them does. Okay, and there is a nice table in your book to help you with this task. So the first question I have for you is, with all of these enzymes, what do they do? And then the second question is, can you articulate why there is a different mechanism used to replicate each strand? Meaning, why is there a lagging and a leading strand? Why aren't both the strands of DNA just copied continuously and simultaneously? Because that would sure make things a lot easier. And then, so explain why that is the case, and then also just describe a little bit about the difference between the leading strand and the lagging strand. Go ahead and pause the video and work through these, and then I'll go through the answers. Okay, so for the different enzymes and proteins, we have 3 different DNA polymerase enzymes. DNA polymerase 3 is the one that builds most of the new DNA, and then polymerase 1 and 2 have kind of some more special functions. So DNA polymerase number 1, this is the one that has that extra exonuclease activity that removes all the RNA primers that are put on to the template strand. Then DNA polymerase 2, this one just kind of has more of a role in repairing DNA that's broken. So maybe not necessarily during this whole process of DNA replication, could be maybe later on when any kind of DNA damage is detected, so on and so forth. And then DNA polymerase 3 is the main one that's going to add new nucleotides to a template strand, and when it's building the new strand is constructed in the 5' to 3' direction. And then just remember, keep in mind, this is a place where people can get confused. It's going to be reading the template strand from 3' to 5'. Okay, so make sure you know if you're talking about which direction it's adding nucleotides or which direction it's reading the template strand, because it's going to travel. If this is the template strand, this is the 3' end, and this is the 5' end, and here's DNA polymerase. Oops. And it's going to bind here and get ready to add a new strand of DNA. Well, that new strand is going to be built in the 5' to 3' direction, but the template strand that it's reading off of, which would be in this little schematic here, here's the template strand, here's the new strand. The template strand is always going to be reading off of the 3' end. Okay, so keep that in mind, knowing the difference between which direction it builds versus which direction it reads from, because that can vary depending on what the question asks. All right, helicase, this is the enzyme that unwinds or it doesn't unwind the DNA, but it breaks apart the double strand by disassociating the hydrogen bonds. Ligase is the thing that's going to seal all the gaps between the Okazaki fragments. Primase, as the name suggests, this is the one that's going to be laying all of the RNA primers. And then these single-stranded binding proteins, these are the things that keep the 2 strands from re-annealing with one another or re-binding with one another after the helicase has kind of come by. All right, and then why is there a different replication mechanism for each strand of DNA? And that gets us to this whole directionality thing. If I'm going to draw a picture of a replication fork, let's say this is these two blue lines represent 2 strands of DNA, and here's the place where they've just been broken apart. Well, if this was the 3' end of one strand, then this would be the 5' end of the other strand, and if that was the case, then this would be the 5' end of this strand, this would be the 3' end of this strand. Well, DNA polymerase, it has to read off of the 3' end of a template and then build in the 5' to 3' direction. Okay, but if I had another DNA polymerase enzyme here, it couldn't read off of here because this is the 5' end. So it has to come over this way and read at it from this direction, because this is going to be where the 3' thing is exposed. So then it attaches here and it builds this way in the 5' to 3' direction. Okay. So the short answer to this long-winded question is there's a different DNA replication specifically because DNA polymerase can only add nucleotides in one direction, and unfortunately for us, the two strands of DNA are anti-parallel, meaning there's two strands running in two different directions. And so if you have an enzyme that can only go in one direction, then it's going to require kind of some clever navigating to make that happen. Describing each mechanism, well, if we're talking about this one here, this would be the leading strand, and this one's pretty simple. Basically, DNA polymerase binds, and it reads the template, and it just continuously adds new nucleotides, and that's all there is to that. The lagging strand is the one that's the hardest to talk about. Okay, so the lagging strand, if this was on an exam or something, you would want to word it something like the lagging strand is built in a series of pieces known as Okazaki fragments. So DNA is going to attach to an RNA primer and build a small segment, and then as this continues to unzip, it's going to bind to another primer that's being laid, and then build another short fragment, and then another short fragment, and then another short fragment. So the lagging strand will be built in a series of short fragments called Okazaki fragments, which are later stitched together by ligase to form a continuous strand. All right, moving on then. So this is how it happens in prokaryotes, and now we're going to talk about how it happens in eukaryotes. But don't worry, the process is nearly identical. So if you're already kind of confused and can't handle anymore, don't worry. There isn't a whole lot of difference between a eukaryote and a prokaryote in terms of how DNA replication occurs. But a couple of the big differences, first and foremost, is that eukaryotes have a much bigger genome. We have about 6 billion base pairs in our genome. So each one of those little nuclei of each little cell has about 6 billion base pairs. Compare that to the 4.6 million base pairs of a bacteria or a prokaryote, and you can see that this is about 1,000 times bigger. Okay, so if you take 1,000 genomes in a bacteria, that's about going to equal what our genome is. And then there's going to be a whole bunch of origins of replication on each chromosome. So remember, prokaryotes have one big circular chromosome, but we have a lot of non-circular chromosomes. We have, for example, in our cells, we have 46 chromosomes, and they are not circular. So there's a whole bunch of different of these origins of replications, and this is, again, a place where replication begins. So if a cell, a eukaryotic cell, for example, is entering the S phase of interphase, and it's going to get ready to duplicate all of its chromosomes, there's just a whole flurry of activity that happens. And there's about 100,000 different spots throughout the entire genome, all of the 46 chromosomes, where DNA replication is then going to start. And then because it's starting in so many places, it can kind of occur much faster than if it were to just start in one place of one chromosome, of each chromosome. That would take forever. Okay, so that's good news for us. There's a whole bunch more DNA polymerase enzymes when we're talking about eukaryotes. There's upwards to about 14 of them versus just 3. And I wouldn't even know what all the difference is between all of those, so I certainly won't ask you about it. And then again, the process is pretty much the same with a couple exceptions. One, the first thing that needs to happen in eukaryotes is that DNA has to unwind from the histone proteins. Remember, in a chromosome of a eukaryotic cell, all of the DNA is wound around these little balls of histone proteins, and that's called chromatin. So a chromosome is literally just DNA that's wound around all of these chunks of protein, and that's just to help it package and condense better. So in order for DNA replication to occur, the chromosome has to kind of get a little bit loose, and all of those little histone protein, they're called nucleosomes, anywhere that a DNA is wound around one of these little balls of protein, they all have to kind of uncoil and unwind themselves so that the DNA is accessible to the different molecular machinery required to replicate the DNA molecule. Another thing that's kind of interesting about eukaryotes is they have these things called telomeres. Okay, and these are kind of bizarre. What a telomere is it's a little region on the end of a chromosome where there's just this repeating phrase, TTAGGGT. And if you were to sequence the series of DNA on the end of a chromosome, all you would see is this repeating phrase, TTAGGGT, or TTAGGG, 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 TTAGGG. It just repeats over and over and over again, and it's nonsense. It doesn't code for anything. And part of the reason it's thought that we have these is because there are, I won't go into it, it's a little bit more complex, but for a reason that's kind of above the scope of this class, chromosomes are always getting shortened. Okay, so if you look, this shows a picture of the end of a chromosome, and every time the cell divides, that chromosome end gets shorter and shorter and shorter and shorter. And the reason being is that at the very end of the chromosome there's an overhang. So if this represents the double strand of DNA, okay, these are the two strands of DNA, there's always a little single-stranded overhang here. There's an enzyme called telomerase which can go ahead and add that little extra piece back so that we have double-stranded DNA all the way to the end. But unfortunately, a majority of our cells do not have this telomerase enzyme, so they're not able to bring that overhang back so that it's double-stranded all the way to the very end of the chromosome. So then what happens is this piece just gets lopped off. And then on the next part, this piece gets lopped off. And then in the next division, the next piece gets lopped off. So lucky for us, though, we have a whole bunch of this kind of just junk DNA sitting here so that as it's getting broken down and shorter, it doesn't actually get into any of the vital genes. Okay? But there's going to come a point at which later in life where it does start to carve into some of the genes that are important. And so that's why telomere shortening has been thought to have some sort of process, is going to be responsible kind of for the aging process. That as those telomeres shorten, they start to get into genes that are useful for regulating certain cellular events. And then if the cellular processes aren't being regulated properly, they're not doing everything that they should be at the right amount of time, then it can lead to some kind of imbalances in homeostasis within the cell, and that can kind of put some strains and stresses on the organism. So there's been some studies to try and link how much of aging is due just to this shortening of telomeres at the end of the chromosome. All right, the next thing that we'll go into is DNA repair, and I think I'll save this for another part of our lecture.